When I introduce this chapter, I like to remind students that everything we see in the human body, every tissue, organ, and its function starts with a single cell. Cell reproduction isn't just about making copies, it's how life renews itself. This process is the foundation for growth, repair, and even how our traits are passed from one generation to the next. Over the next couple of slides, we'll cover the story of DNA's discovery and how cells divide, differentiate, and specialize to form an incredible diversity of structures in the body. I also want you to think of this as the chapter where chemistry becomes life in motion, where molecules translate into living functions. Let's start by thinking about what makes life possible at the most fundamental level. Every cell in your body, from muscles to neurons, runs on the same set of molecular instructions, and that is known as DNA. In 1953, James Watson and Francis Crick described its now famous double helix structure, which is a shape not only defines biology but reshapes how we think about its inheritance. DNA carries all of the copied information that directs how an organism grows, functions, and even behaves. It's the blueprint for every biochemical and anatomical feature we have. What's fascinating is that all of this complexity, your organs, tissues, and even thought, comes from how DNA is read and expressed within each cell. This sets the stage for the understanding of how cells reproduce, specialize, and repair the body. Before we understood DNA's role, many scientists believed proteins carried heredity because they were more complex. But a series of experiments changed that. T.H. Morgan's work with fruit flies showed that genes reside on chromosomes, which contain both DNA and proteins. Then Frederick Griffith's famous 1928 experiment with harmless and pathogenic bacteria showed that some transforming principles could pass traits from dead cells to living cells. This transformation meant that something physical, later revealed to be DNA, carried genetic information. Avery, McLeod, and McCarty confirmed in 1944 that DNA was transforming substance, not proteins or RNA. Soon after, Alfred Hershey and Martha Chase used bacteriophages, which are viruses that infect bacteria, to settle the debate. By labeling DNA and proteins with radioactive tags, they showed only DNA entered the bacterial cells during infections. This proved that DNA, not proteins, directs inheritance. What's fascinating here is that these monumental discoveries came from not from humans or animals, but from other little tiny microbes. As researchers on DNA advanced, Erwin Chargaff made discoveries that changed everything we thought we knew about genetic material. He compared DNA samples from different species and found two key things. First, the ratio of bases varied depending on the organisms, which explained why DNA could carry unique genetic information. Second, within a single species, the amount of adenine always equaled thymine, and the amount of guanine always equaled cytosine. These became known as Chargaff's rule. At the time, no one knew why these ratios mattered, but later they became the missing puzzle piece for Watson and Crick. His work hinted that the bases might pair in some consistent way. Rosalind Franklin's work marked the turning point in understanding DNA structure. Using a technique called X-ray crystallography, she captured diffraction images that revealed DNA's repeating helical pattern. Her most famous image, known as Photo 51, showed the distinctive X-shaped pattern that could only come from a helix. This photograph showed scientists to calculate the width of the molecule and the spacings between the stacked bases. What's remarkable is how much precision and patience this work required. She didn't have a computer or digital imaging. She used mathematical interpretations of scattered X-rays. Her data provided the critical evidence that DNA was a double helix composed of two strands winding around each other. Franklin's contribution often went unrecognized at the time, but today we know her meticulous science made the double helix model possible. Once Rosalind Franklin's data confirmed that DNA was helical, Watson and Crick began building physical models to understand how its components fit together. They discovered that DNA structure had two sugar phosphate backbones running in opposite directions, what we call the antiparallel strands. These backbones form the outer edges of the helix, while the nitrogenous bases are paired in the interior like rungs on a twisted ladder. At first, they thought Bases might pair randomly, but they didn't work geometrically. The breakthrough came when they realized a purine, which is a larger, must pair with a pyrimidine, which is smaller. This pairing, adenine and thymine and guanine with cytosine, created a uniform width throughout the molecule and perfectly matched Franklin's data. The beauty of the structure is that it explains its own function. Each strand can serve as a template for copying, ensuring the genetic code is passed down in remarkable accuracy. Now that we've covered DNA structure, let's look at what cells actually do with it. Every multicellular organism begins as a single fertilized egg, and through countless divisions, it develops into a complete body with trillions of cells. For humans, that one zygote becoming around 10 trillion specialized cells. Cell division is the engine behind this growth, repair, and replacement. But division alone isn't enough. After cells divide, they begin to differentiate, meaning they take on specific roles. A neuron, a muscle cell, and a skin cell all share the same DNA, but they express different parts of it. The cycle is the repeating pattern of growth and division that defines cellular life. Most of cells are spent in interphase, the long stretch between divisions. During G1, or the first gap phase, the cell grows, carries out the normal duties, and prepares for DNA replication. The S phase, short for synthesis, is when DNA is copied. Every chromosome is duplicated. Then comes G2, the second gap phase, when the cell makes final preparations for division. Think of interphase as a quiet but essential part of the cycle. Once interphase is completed, the cell enters the mitotic phase, when all the preparation pays off. Mitosis divides the nucleus, ensuring that each daughter cell receives an identical set of chromosomes. Cytokinesis follows, splitting the cytoplasm and producing two distinct cells. Together, these processes take less than an hour, which is incredibly fast considering the complexity involved. Not every cell keeps dividing forever, though some, like neurons and muscle cells, enter a permanent resting phase called G0, where they stay functional but no longer replicate. Others, like skin and intestinal cells, divide constantly to keep tissues renewed. A typical human cell takes about 18 to 24 hours. The majority of that time is in interphase, while mitosis and cytokinesis makes up only a tiny fraction. Timing is everything here. If a cell divides too quickly or too slowly, it can cause developmental issues or disease. The body uses checkpoints to make sure each phase is completed before moving to the next. This precision ensures that cells don't divide until DNA is fully replicated and any errors are fixed. It's the biological equivalent to a security system. Now that we've talked about what happens when cells divide, let's revisit what they're dividing. Each human cell contains about 3 billion base pairs of DNA organized into 46 chromosomes. These chromosomes consist of DNA wrapped around histone proteins, forming a complex called chromatin. Most of the time, chromatin is loose and thread-like, allowing genes to be accessed and transcribed. But during cell division, it condenses into visible chromosomes so it can be distributed precisely. Each chromosome consists of two sister chromatids joined at a centromere. Altogether, our genome contains about 20,000 to 21,000 genes. Each one has a short segment of DNA carrying the code for a protein. This is the foundation for everything that happens next, from replication to gene expression. DNA's job isn't just to exist, it's to act. These are three essential processes that bring genetic information to life: replication, transcription, and translation. Replication is how DNA copies itself before the cell division, guaranteeing that each new cell inherits the same genetic instructions. Transcription is when a section of DNA is converted into RNA, a working copy that carries information out of the nucleus. When translation takes that RNA code and uses to build proteins in the cytoplasm. These proteins become enzymes, hormones, and structural components that make life possible. Together, these three processes form the central dogma of molecular biology. DNA makes RNA, RNA makes proteins, a seamless flow from information to action. Replication isn't one of the most elegant molecular processes in DNA. Before a cell divides, it must duplicate all 46 chromosomes, roughly 6 billion base pairs. The DNA molecule unwinds like a zipper, separating into two separate strands. Each strand then serves as a template for a new complementary strand. Adenine pairs with thymine, and cytosine pairs with guanine, ensuring perfect fidelity. The enzyme DNA polymerase builds these new strands, checking for errors as it goes. It's mind-blowing how accurate this process is. Only about one mistake per billion nucleotide copied. Of course, replication isn't perfect. Sometimes errors slip through, or external factors like radiation, chemicals, or UV light damage the DNA. These changes are called mutations, alterations in the nucleotide sequence. Some mutations have no effect, others can cause disease, and a few drive evolution by introducing new traits. In body cells or somatic cells, mutations can contribute to cancers or tissue malfunctions but aren't passed to offspring. In gametes, though, sperm and eggs, mutations become part of the genetic legacy, passed on to the next generation. Fortunately, all cells aren't passive when damage occurs. They're equipped with DNA repair enzymes that constantly patrol the genome. When a mismatch or break is detected, the damaged section is cut out, replaced by new corrected nucleotides that seal back together by repair enzymes. This repair actively peaks between DNA replication and mitosis, which is a critical window before cell division. Mutations in the gene that code for these repair enzymes can cause cancer risks since errors begin to accumulate unchecked. Now we turn to how DNA's code is read and used. In transcription, a specific gene is copied into the messenger RNA, or mRNA, inside the nucleus. This mRNA acts like a portable version of the genetic recipe. It then travels to ribosomes in the cytoplasm where translation takes place. During translation, the sequence of mRNA codons, group of 3 bases, directs the assembly of amino acids in the protein chain. Together, these steps transform the language of nucleic acids into the language of life, proteins. Transcription is the first step in turning genetic information into something useful. Inside the nucleus, the DNA of a single gene unwinds, exposing the base sequence. An enzyme called mRNA polymerase moves along the DNA, matching complementary RNA nucleotides to the exposed bases, forming a strand of messenger RNA or mRNA. At first, this mRNA copy includes both useful and non-useful sections, the exons and introns. Introns are removed by special enzymes called ribozymes, leaving only the exons that carry the instructions for making proteins. What remains is a clean functional mRNA molecule ready to leave the nucleus. Once mRNA is made, it carries instructions within the 4 letters alphabets of RNA, A, U, G, and C. Each word in the codon consists of 3 bases called codons. Each codon specifies 1 amino acid. There are 64 possible codons, but only 20 amino acids, which means that the genetic code is redundant. Multiple codons can specify the same amino acid. For instance, GAA and GAG code for glutamic acid. This redundancy helps protect against minor mutations. The code is read in one direction, from 5' to 3' of the mRNA strand. The triplet system ensures that proteins are built accurately, 1 amino acid at a time. By the mid-1960s, the scientists had deciphered all 64 codons, 61 codons for amino acids, and 3 functions to stop signals that end translation. What's extraordinary is that this same codon is shared nearly by all organisms, from bacteria to humans. This near universality suggests that the genetic code evolved early and has been preserved for billions of years. It also means genes from one species can be expressed in another, the principle behind genetic engineering. For example, we can insert the human insulin gene into bacteria and they'll produce insulin identical to our own. The genetic code has its own grammar. Translation begins with a start codon, AUG, which codes for the amino acid methionine. This signals the ribosomes to start building the proteins. The process continues until one of the 3 stop codons tells it to end. These codons don't code for amino acids. Instead, they act like punctuation marks at the end of a sentence. This organization ensures that proteins are built to the correct length and sequence. If the ribosome reads the code out from frame, even one by one, the entire message changes, often with disastrous results. Here we see the central dogma of biology in action. DNA makes RNA, and RNA makes proteins. In prokaryotes, this process happens quickly and efficiently because transcription and translation can occur simultaneously. There's no nucleus separating the two. In eukaryotes like us, transcription happens inside the nucleus and translation takes place in the cytoplasm. This separation allows more control and modification of RNA before it is used. The mRNA that leaves the nucleus is polished version of the original transcript. This flow of information from DNA to RNA to proteins is what connects genotype to phenotype, turning genetic information into structures and functions of the human body. Now that the mRNA blueprint is ready, translation is where the magic of proteins builds happens. The main players are three types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. The mRNA brings the coded instructions from the DNA to the nucleus to the ribosome in the cytoplasm. The ribosome, built partly of rRNA, serves as the workbench where the protein is assembled. Finally, tRNA molecules act as carriers, each carrying a specific amino acid and matching the anticodon to the correct codon on the mRNA strand. When all of these pieces come together, amino acids are linked in precise order, forming the proteins that perform almost every job in the cell. Enzymes, hormones, transporters, you name it. Translation unfolds in three stages: initiation, elongation, and termination. During initiation, the ribosomes attach to the start codon of the mRNA, and tRNA carrying methionine binds to and begins the chain. During elongation, more tRNAs bring amino acids to the growing protein, linking them one by one through peptide bonds. These ribosomes move along the mRNA like a molecular assembly line, translating each codon into amino acids. When a stop codon appears, the process halts and the completed protein is released. The new protein will then fold into a functional shape, sometimes immediately, sometimes with help from other cellular molecules like chaperones. With DNA and protein synthesis covered, let's return to the big picture: how cells reproduce. There's two main types of cell division: mitosis, which produces new diploid cells genetically identical to the parents, used for growth and tissue repair, and meiosis, on the other hand, which creates haploid gametes, which are sperm and egg cells carrying only one set of chromosomes. This reduction is crucial for sexual reproduction because it ensures that when two gametes fuse together during fertilization, the resulting zygote restores their full diploid number. The two processes, mitosis and meiosis, balance continuity and diversity. Mitosis is the process that ensures every new cell gets a complete and identical set of genetic instructions. It's a form of nuclear division, following by cytoplasmic division, or cytokinesis. Together they allow one parent cell to produce two genetically identical daughter cells. What's important is that mitosis does not just make more cells, it maintains genetic continuity, so your new skin cells or bone cells have the exact same DNA as the originals. The process unfolds in four major stages: prophase, metaphase, anaphase, and telophase, each with precision and molecular choreography. You can think of mitosis as a highly rehearsed dance where timing and structure are everything. Mitosis begins with prophase, when the chromatin, that loose stringy DNA, coils tightly into a visible chromosome. Each chromosome is made up of two identical sister chromatids connected by a central mere. The mitotic spindle form is made up of microtubules that stretch between the centrosomes at opposite poles. The nuclear envelope breaks down, signaling the start of active division. Then we move into metaphase, where all of the chromosomes line up in a single plane along cell's equator, called the metaphase plate. This perfect alignment ensures that when the chromosomes separate, each daughter cell will receive one full set. If there's even seen cells under a microscope during metaphase, the neat line of chromosomes looks like the cells holding its breath, waiting to split. During anaphase, the paired chromatins separate, now becoming individual daughter chromosomes. The spindle fibers shorten, pulling towards opposite poles of the cell. It's the moment of separation when each side gets an identical set of genetic instructions. Then comes telophase, which is essential for reversing of prophase. The chromosomes begin to uncoil back to chromatin, nuclear envelope reforms around each set of chromosome, and the mitotic spindle dissembles. At this point, two new nuclei are visible within the same cell. The genetic division is complex. Now the cell just needs to divide physically. Telophase marks the calm after the storm, the final step before the cell truly becomes two. Cytokinesis is the process that physically separates the cytoplasm into two daughter cells. In animal cells, a contractile ring made of actin filaments tightens at the center of the cell, creating what we call a cleavage furrow. This furrow deepens until the cell is pinched in half. Each new cell receives not just a nucleus, but also a fair share of organelles, cytoplasm and nutrients. The result, two genetically identical diploid cells that ready to enter the interface and begin their own life cycles. This happens constantly throughout our body, in your skin, your intestines, your bones, keeping tissues alive and it's functional. All somatic cells or body cells in the human body divide by mitosis, ensuring that each new cell has a complete and identical set of chromosomes. Every diploid cell contains 46 chromosomes arranged in 23 pairs. These pairs are known as homologous chromosomes, one inherited from the mother and one from the father. Together, they carry the matching genes that govern the same traits, although the versions of these genes, the alleles, may differ. Out of the 23 pairs, 22 are autosomes, which determine most of the body's traits, and one pair of sex chromosomes. Females have 2 X chromosomes, while males have 1 X and 1 Y. This chromosomal organization is what maintains genetic stability from one cell generation to the next. The only cells that don't follow this pattern are sperm and egg cells, which are produced by meiosis, a process designed to cut the number in half so fertilization restores to 46. Gametes, which are sperm in males and eggs in females, are unique because they are haploid. That means they contain only one set of 23 chromosomes instead of the usual 46. This reduction is critical. Without it, fertilization would double the chromosome numbers with each generation. This process that makes haploid cells in meiosis a specialized type of cell division that takes place only in the ovaries and the testes. During meiosis, one diploid cell divides twice, producing 4 genetically distinct haploid cells. This reduction from diploid to haploid ensures that when a sperm and egg unite to fertilize, the resulting zygote has the correct diploid number of 46 chromosomes. So while mitosis creates identical cells for growth and repair, meiosis ensures that genetic diversity and continuity across the generations. Meiosis is a 2-part division process that transforms one diploid cell into 4 unique haploid cells, which with half of each chromosome number. The first division, meiosis 1, separates the homologous chromosome pairs and includes the stages prophase 1, metaphase 1, anaphase 1, and telophase 1, as well as cytokinesis. The second division, meiosis 2, closely resembles mitosis with its own prophase, metaphase, anaphase, and telophase phases, followed by cytokinesis again. The key difference is that meiosis cuts the chromosome number in half, a process we call reduction division. The result is 4 genetically distinct daughter cells, each haploid containing 23 unpaired chromosomes. When fertilization occurred, the haploid sperm and the haploid egg combine to restore the diploid number of 46 chromosomes in the zygote. Meiosis 1 is where real genetic mixing happens. It begins with prophase 1, when homologous chromosomes, one from each parent, comes together and pairs up closely. This pairing allows them to exchange segments of DNA in a process called crossing over. This exchange creates new genetic combinations, ensuring that no 2 gametes are ever identical. During metaphase 1, these chromosomes paired line up side by side at the equator of the cell, forming a distinctive double line, a key feature that distinguishes meiosis from mitosis. In anaphase 1, the homologous pairs separate and move towards the opposite poles, but the sister chromatids remain attached. Finally, in telophase 1 and cytokinesis, the cell divides into 2 daughter cells, each with half of the number of chromosomes, now haploid. However, the chromosomes are still duplicated, each consisting of 2 chromatids. This sets the stage for mitosis 2. Meiosis 2 begins with 2 haploid daughter cells produced by meiosis 1. Each one now goes through a second division that closely resembles mitosis. During prophase 2, the chromosomes condense again and a new spindle fiber forms in each cell. In metaphase 2, the chromosomes line up on a single file along the center equator, not in pairs like before. Then comes anaphase 2, where the sister chromatids finally separate and are pulled to opposite poles of the cell. This is where the full reduction to a single chromatid happens. During telophase 2 and cytokinesis, the nuclear membrane reforms and the cytoplasm divides. The final result is 4 non-identical haploid cells, each containing 23 chromosomes. These will develop into gametes, sperm or egg, that carry unique genetic combinations. Meiosis 2 is the final step to ensure genetic variety, which is why no two siblings except for identical twins are ever genetically the same. Meiosis works a little differently in males and females, even though the overall purpose is the same, to create haploid gametes. In male, each cell that enters meiosis produces 4 sperm cells, and all 4 are viable and functional. This efficiency is key for reproduction because millions of sperm are needed to increase the odds of fertilization. In females, however, meiosis is unequal. Cytokinesis divides the cytoplasm unevenly, producing 1 large egg cell and 3 much smaller cells called polar bodies. The egg remains nearly all of the cytoplasm, resources that will sustain the embryo after fertilization. The polar bodies, which contain chromosomes but little else, eventually degenerate. This difference ensures that the egg is well equipped to support early development, while sperm are optimized for quantity and mobility. Not all cells in the body divide the same rate, and that's because cell reproduction is tightly controlled by an internal surveillance system. The cell cycle control system acts like a series of checkpoints at G1, G2, and M, where the cell must receive a go-ahead signal to continue. These checkpoints ensure that DNA is correctly replicated, that the cell is large enough, and that the environment is suitable for division. If any of these conditions aren't met, the cell pauses for repair or even triggers programmed cell death, known as apoptosis. External factors such as hormones, growth factors, and contact with neighboring cells can also influence when a cell divides. For instance, skin cells divide more rapidly when healing a wound, while nerve cells stop dividing altogether after adolescence. This precise regulation keeps tissues healthy and prevents uncontrolled growth, the kind that leads to cancer. Every cell in our body contains the same DNA, yet they don't all look or act alike. That's because differentiation. Differentiation is the process by which cells become specialized in their structure and function. This process depends on which genes are turned off or off in each cell. Environmental signals like hormones, nutrients, and neighboring cells influence how a cell interprets the genetic instructions. For example, a stem cell can become a muscle cell, neuron, or skin cell depending on the environment and chemical cues. This selective gene expression gives rise to the diversity of tissues that make up the human body. It's a powerful reminder that DNA provides the instructions, but the environment determines how the instructions are read. Differentiation begins soon after fertilization. Once the sperm and egg fuse to form a zygote, that single cell starts dividing rapidly. But at first, these divisions don't increase the embryo's size. By the 8-cell stage, each cell begins to experience a slightly different environment, depending on its position within the embryo. These cells' differences triggers the first steps towards specialization. Before this point, all cells are identical and can still give rise to an entire organism. This is why embryo splitting can produce identical twins. However, after about the 16-cell stage, these cells become committed to certain fates, and cloning by splitting is no longer possible. It's an elegant transformation from potential to purpose. As the embryo continues to grow, differentiation becomes guided by 2 main factors: a cell's developmental history and its local environment. Certain genes are switched on or off depending on where the cell is and what signals it receives. This is why cells in the heart become muscle cells and those in the brain become neurons. The timing of gene expression is crucial. Damage to DNA early in development can cause miscarriages or severe birth defects, while mutations later may affect only one tissue. In adults, some mutations go unnoticed, while others can trigger diseases like cancer. Differentiation, therefore, is a long life balancing act between stability and adaptability. External factors can seriously interfere with normal development, especially during pregnancy. Substances like cigarette smoke can restrict oxygen and stunt fetal growth. Alcohol can cause fetal alcohol syndrome, leading to brain and facial abnormalities. Medications and illegal drugs can cross the placenta, affecting fetal brain chemistry or leading to dependence at birth. Environmental chemicals in air, water, and soil, such as heavy metals or pesticides, can also disrupt development. Even radiation from medical imaging or natural sources like radon poses risks. Certain infections, including HIV, syphilis, rubella, and Zika virus, can pass through the placenta and cause serious defects. This final reminder ties the chapter together. Cell division and differentiation are precise, but they're also vulnerable. Protecting these processes is essential to ensuring healthy growth and development.